Hello, everyone, and thank you for joining our first bite-sized science virtual seminar of 2021, Harnessing the Immune System Against Pancreatic Cancer. We hope you all had a wonderful and restorative summer, albeit with some COVID-19 disruptions, and that you're enjoying a positive start to the new year. For those of you who don't know me, my name is Mara Jean Tilley, and I have the great privilege of heading up the Garvan Research Foundation, which is the fundraising and marketing arm of the Garvan Institute. And I thank all of you for coming today. I know that we've got some long-standing family members of the Garvan, and I think we've got some new faces as well. So great to have you all in the Zoom room. Before we get started, I'd like to acknowledge the Gadigal people of the Eora Nation, the traditional custodians of the land on which the Garvan Institute and St Vincent's Hospital Precinct are located, and pay my respects to elders past, present and emerging. A big thank you to everybody who attended our bite-sized seminars in 2020 and who gave really valuable feedback to how we can improve these online events. For now, we will be continuing to meet in the Zoom room, but we do hope, forgive me, that by the end of the year, or as soon as it's safe to do so, we'll be able to begin commencing on-site in-person virtual seminars, as well as continuing the virtual seminars, so that for those individuals who live outside of Sydney or interstate or indeed overseas can continue to join us. So for today's topic, the pancreas is an organ that lies behind the lower part of the stomach. It secretes enzymes that help to break down sugars, fats, starches, and hormones to help regulate blood sugar levels, appetite, and stimulate the stomach's acids. Pancreatic cancer occurs when cells in the pancreas develop mutations in their DNA that can cause these cells to grow into tumours. Pancreatic cancer is difficult to detect in its early stages, with patients often going undiagnosed until the tumour has spread to other parts of the body, which makes it very, very difficult to treat. Accordingly, it's the fourth leading cause of cancer death in Australia and has a shocking 5-year survival rate of only 9%. That's a small improvement from 7% some years ago when I started at Garvan, but still deeply inadequate. Today, we've got the honour of hearing from Associate Professor Marina Pajic, Head of the Personalised Cancer Therapeutics Lab here at the Garvan Institute, and renowned and a real superstar leading talent in this area. In Marina's latest study, she's using a new world-first intravital imaging technology to directly see into the dark spaces of pancreatic cancer tumours, in the hope that we can, through this new technology, improve outcomes in immunotherapy to give pancreatic cancer patients a better chance at a longer life and to improve their quality of life. Today's format will include a poll at the conclusion of Marina's presentation for you to vote on questions. If you find that you've got questions that have not been addressed throughout today's presentation, please do take the time to submit your question with the poll at the very end, along with your email address, so that the team can come back to you one-on-one and give you the answers you seek. It's now my great pleasure to introduce Associate Professor Marina Pajic. Welcome, Marina. So tell me, this is an exciting time for you and the team. You've got this brand new technology that's not available anywhere else in the world. Congratulations. And I also believe that you're in the development phase of setting up a clinical trial for pancreatic cancer that's all going well. Will be going live in 2022. Can you tell us a little bit about that, please?
Well, I will tell you all about it in my talk, actually. So yes, both things are very exciting for us. So firstly, I was part of the team who has secured funding from the ACRF, the Australian Cancer Research Foundation, and Cancer Institute New South Wales support as well, to establish this advanced microscopy centre. It's going to be state-of-the-art microscopy, really, to allow us to look much faster, much deeper inside the organs, the live organs, and study how certain drugs work, how they work in the context of the immune system, and then also try to use this to try to figure out why pancreatic cancer is so resistant to immunotherapy. And then the second one, again, I will touch on that during the talk, is a Cancer Institute New South Wales translational program grant that Paul Timpson, myself, really in collaboration with Professor David Thomas and the Most Clinical Trials Program, as well as Redex Pharma. It's a UK company. We have funding now to run a trial in pancreatic cancer. It's based on precision medicine principles. It's a tumor stroma co-targeting approach, but I'll talk about that.
Fantastic. Well, in that case, over to you. Thanks, Marina.
Well, thanks very much for being here today. And thank you for that lovely introduction, Mara Jean. I will just share the slides. All right. Okay, well, without further ado, I will tell you firstly a little bit about why I am passionate about research into pancreatic cancer. And Mara Jean has already covered that quite a bit. So firstly, just to give you a few numbers, just this year, over 430,000 people globally will die from pancreatic cancer, unfortunately. And this is only 3 to 6 months post-diagnosis. 5-year survival rates have remained stubbornly low. They're only sitting at about 9%. It's actually currently estimated as the 3rd leading cause of all cancer deaths in Australia. So these are the latest Cancer Australia figures. And the current treatment in the form of chemotherapy, things like gemcitabine combination, gemcitabine Abraxane, or FOLFIRINOX, really only improves survival by a few months when given to all comers. It's unfortunately still an underfunded and understudied malignancy. So my group really focuses on trying to understand why pancreatic cancer is so resistant, and then trying to define new treatments, more tailored treatment combinations for the disease. Now, this slide, you don't really need to memorize, but it's essentially a summary of the genome sequencing studies that we have performed by sequencing over 400 human pancreatic cancers. And essentially what we found was that pancreatic cancer is molecularly or genetically very diverse. But if you look at the gene networks, so how these genes interact and work together, and that's kind of summarized here on this right circular plot, is that you can begin to see at least 10 molecular or genetic subtypes of pancreatic cancer. So that's the analysis of the tumor cells themselves. But if you look at pancreatic cancer under the microscope, the story actually gets even more complicated. So just showing you what the normal pancreas looks like on the left, you can really see nicely organized structures. There are cell types with specific functions, and everything works, everything works well and in unison. Now, if you look at pancreatic cancer, it actually looks nothing like the normal pancreas. So firstly, you have these really disorganized cancer cells, so highlighted here within this purple border. Cells are stained blue. And then what you have here is this tumor environment or tumor microenvironment. So this contains this really fibrous matrix. It's almost looks like a scar tissue. It's very firm, very stiff. And so within that matrix, it also contains specialized cells called fibroblasts, which make this matrix. Collagen is one of the key proteins within this matrix. And then it also has some of the other cell types, things like blood vessel cells, immune cells, and others. So when we look at, when we're looking at designing new treatment strategies, what's increasingly becoming obvious to us is that we not only have to consider the tumor cell genotype, but we should be looking at the environment that surrounds these cancer cells. And so that's what my lab is focused on. To do this, we are very fortunate because we were part of this large international cancer genome consortium study. So as part of that, we have the genomic information on a large number of pancreatic cancers, but also associated with that, we've also made models of the disease. So these are quite unique patient-derived tumor models. We've got patient-derived cell lines that we use routinely for 2-dimensional as well as 3-dimensional drug screening. we can then take the most promising therapies into our mouse models. So these are individual tumors grown in mice. We can then perform biomarker-driven treatment testing. And associated with that, we do a whole heap of imaging as well to study how these drugs work inside the tumors. And then associated with the genome sequence information, we also have large patient cohorts that enable us. So these are well-annotated clinical cohorts that really allow us to perform our biomarker discovery and as well as validation work. So can any of these subtypes, be targetable? So that's actually what we investigated a couple of years ago now. So one of these gene networks called the cell cycle network or cell division network was of particular interest to us because it had agents available that are used in the clinical treatment of specific types of breast cancer. So we wanted to look at whether we could repurpose in particular this agent called palbociclib, or you can see the long chemical name, sorry, not chemical, but company name here. It's called PD0332991. We wanted to see whether we could repurpose it to treat specific subtypes of pancreatic cancer, specifically those that have this cell cycle or cell division pathway aberrant. And then the first step was in order to look at cell division, we introduced this really nice Fuji, it's called the Fuji probe. We also call it the traffic light probe, because just like the traffic light, you can insert it into the cells and then you can look at how the cells respond to treatment. So if it's red, if the cells go red, that means they stop dividing or stop growing. When it's yellow, that means that those cells are actually probably not responding to treatment as well, and they're getting ready for cell division or cell growth. And green means go, right? So the cells are actively dividing. So when you just have a two-dimensional culture of all this kind of mixed bag of cells, you can see that you've got green cells, red cells, yellow cells. They're really just kind of growing and dividing in culture uninterruptedly. What's quite striking is that when you put a drug on like palbociclib, specifically on cells that are defined by biomarkers, which I'll mention in the next couple of slides, is that over a period of 24 hours, you can see that cells become red. So they stop growing effectively. And then you can take this in vivo. So you can look at how tumors actually respond to this agent. And so this is really when things start to get interesting, because in two-dimensional culture, you're really only looking at the effect of your targeted agent on cancer cells. But as I mentioned before, environment plays an important role also. So we should look at the effects of these targeted drugs and see what they do to the tumor microenvironment. And so again, here you can see the PD0332991 treated tumor. All the tumor cells have gone red. So it's effectively blocking cell growth or cell division in these tumors. What we also then looked at, so this is shown here in purple, is this scar tissue, and particularly one marker called collagen, which is the predominant protein present in this scar tissue surrounding these cancer cells. And this just shows you a cross-section of a tumor region where you can see that in the presence of this drug, so this was quite unexpected for us, that this targeted agent was actually able to significantly reduce the scar tissue surrounding this particular cancer type. And so what we know about the scar tissue is that, it plays many functions in promoting tumor cell growth and tumor cell development. But at the same time, it also effectively, because it's quite stiff, it provides a nice structural matrix for the tumor cells to attach to grow and be comfortable in. It also directly blocks the permeability to cancer agents. So if you can modify this scar tissue or try to reduce it somewhat, there's potential to then improve response to chemotherapy. And so then when we looked at this, so these are long-term follow-up studies in using these in vivo models. What you can see here, you don't need to specifically worry, but like we have about these pictures here, but we've actually been able to tailor response according to specific biomarkers, right, present in these tumors. So RB was a key determinant of responsiveness to the palbociclib-based monotherapy and combination therapy treatment. And that's just stained here. So these tumor cells are staining bright brown for this particular protein. And importantly, what we've shown is that when you treat these tumors, you can, with palbociclib or palbociclib-based clinically relevant combinations, this would lead to over 3 times improvement in survival over, chemotherapy alone. So this is again gemcitabine alone in blue, or gemcitabine Abraxane combination as 2 standard treatment approaches. So that was quite promising to us. Since then, we've also been really interested in other potent modulators of this stromal or tumor microenvironment. And so this just shows you another vignette of how imaging can help you really layer and look at the sort of multifaceted effects of some of these agents. And so this particular agent was a small molecule inhibitor of a very important kinase, very important protein that regulates the how cancer cells migrate or move, how they attach to the matrix, and also how they grow. So it's called ROCK. And so we're using ROCK inhibitors. This was an old drug that we've used here, a ROCK inhibitor called Fasudil. And again, looking at the presence of this scar tissue in these tumors post-treatment. And what's quite striking is that, in comparison to our control tumors and the gemcitabine Abraxane, so this is just standard chemotherapy treated tumors, the ROCK inhibitor-based either monotherapy or combinations really dramatically reduce the presence of collagen in these tumors. But what's really beautiful about intravital imaging is that in addition to this, it really enables that, you can look at multiple different layers in a single, animal in live tumor tissue. So here I'm just showing you, so this is just an untreated tumor, but it just illustrates, we've got cancer cells, pancreatic cancer cells that will light up in green. Again, purple is the scar tissue. And then we've also injected these quantum dots. So this is a red label that will pop up here that labels the vasculature of these tumors. So it looks quite striking as you go through the cross-section of the tumor. You can really now start to appreciate the complexity of, these growing pancreatic tumors. And what gets even more exciting is when you start to look at these tumors in the presence of different agents. So this is the effect of the ROCK inhibitor. Now we're going to be looking at the vasculature of these pancreatic tumors. So we've got blood vessels here highlighted in the red. And then what we became interested in is the perfusion, really. So how many of these little dots, red dots, are capable of leaving the blood vessels and permeating the tumor tissue. And so if I just turn the video on, you can see that in comparison to the control tumors, which are really kind of, the vasculature is well defined, when you treat tumors, so when you treat these models with Fasudil, the ROCK inhibitor, the blood vessels really become more leaky. And so these little dots permeate the tumor tissue much more. And what that's signaling to us is that this is likely to be happening when you now, on top of this, treat with chemotherapy, it's more likely to get into the tumor itself, so to the tumor cells, right? And hopefully kill the tumor cells. And this just provides you another view. So it's just a 3-dimensional cross-section. So it just allows you to appreciate more the sort of the kind of permeable or the perfuse areas in the tumor, in the pancreatic tumor tissue once you treat with Fasudil, the ROCK inhibitor. And that again leads to, when you look at survival, it leads to dramatic improvement in both survival as well as sensitivity to chemotherapy. So that's just highlighted here in red. And really comparing it to chemotherapy alone, gemcitabine shown here in blue. You've got the control, so that just saline treated animals in black. So you really see the lack of effect of chemotherapy in specific tumor types. And then you can also see how dramatically you can really begin to improve this if you target specific mechanisms with targeted therapies. And so that leads me to what is now actually, hopefully it's not so much the future relevance, but hopefully near future relevance or current relevance for precision medicine in pancreatic cancer. So this is the trial that we're hoping will start mid this year. As I said, this is a collaboration with Professor David Thomas and MOST program, RedX Pharma, and also Paul Timpson and my group as well. And so this is what we're doing here. We'll be taking patient tumor material, performing sequencing, so this is panel sequencing analyses to identify specific mutations, and alongside that, also profiling the fibrotic nature of pancreatic cancers. So we'll be looking at targeting highly fibrotic tumors, as well as we're looking at specific mutations in the tumor cells to target with a selective dual inhibitor of both porcupine signaling and ROCK signaling. So it's a selective drug. It's early in clinical, I shouldn't say it's not super early in clinical development. It's past phase 1 safety testing. So this will now be a phase 2 biomarker-driven precision medicine-based clinical trial. And then we're hoping to do a lot of correlative science on the tumor material bloods that we'll be collecting from these patients to really try to figure out and refine potential signatures behind treatment response as well as treatment failure. So because I promised that I was going to talk a little bit about the immune system, just in the last couple of slides, I'll tell you a little bit about the difficulty with targeting the immune system in pancreatic cancer. So the immune system, is quite complex, but it's very effective early on in controlling early tumor cell growth. So as we age, cells in our body they constantly accumulate mutations. And so you will get occasional rogue cells that then the immune system can really effectively block and get rid of so that they never become cancer. But what happens eventually is that at some point you get these little selected tumor cells that develop specific mechanisms so they can evade the immune system. And they can sit like this quite quietly for a number of years, evading the current immune system, and then also acquiring additional mutations that will help them develop additional mechanisms to really become resistant to any sort of immune targeting. Until eventually they lead to this phase called the escape phase, where you get this uncontrolled tumor growth. You then also get shutdown of these, what we call the good guys, sorry, excuse me, which are the CD8 positive T cells, which are able to recognize and kill cancer cells normally. So they get shut down. The tumor cells further recruit additional immunosuppressives. So these are additional immune cells that further dampen the immune response. And that really allows the cancer cells to overtake and grow. And so unfortunately, pancreatic cancer is usually diagnosed at this escape phase. So what my team is really interested in is trying to wake up the immune system. So we're looking at many different approaches. We're looking at novel agents, but just to show you some of the data here, we're also very interested in repurposing some of the non-oncology drugs as pancreatic cancer therapy. And so what we found here is that one of the really old drugs called itraconazole, it's an antifungal agent. It's used to treat fungal infections. It's very safe and tolerable. What we found is that in pancreatic cancers, it actually decreases so it blocks the bad guys. It decreases these F480 positive. So this is this brown staining shown here within these pancreatic tumors. So it's reducing the load of the bad guys, and then simultaneously is beginning to wake up the good guys in these tumors. And so what we think is particularly for us is hopeful is that we can maybe look at how we can switch on the immune system and then really stimulate the response of pancreatic cancers to something like immunotherapy, right? Because when immunotherapy works, it's really, truly amazing. Now, these pictures that I've shown you here, the problem is that these are static analyses, right? So its immune system is very fast and dynamic, and this is just a snapshot at a specific time point. And so just to show you one last video here is, so this is what it looks like. And so this is a specific population of cells. Now it's the neutrophils, the white blood cells, how they're invading a wound. Again, we've got purple as collagen staining, and this is just a superficial wound in the ear. So what you can hopefully begin to appreciate is that, you know, although this is, these are quite pretty and beautiful videos, we need to go deeper. We need to go into the pancreas, we need to go into the liver. These are deep within the abdominal cavity, so much harder to image, and we also need to image faster. And so this is why the now funded ACRF Insight Center is particularly important for our future work. And that's really what we're hoping to achieve. And so just a few points to leave you with. The future of personalized medicine is here. We're working on it. So are many others around the world, but it's looking hopefully promising. We are very keen on building more effective combination treatment strategies. So that's really something that we're working on actively. We are considering the immune system. We have to look at the whole tumor environment surrounding these tumor cells, not just the cancer cells themselves. And then some of the other points that we're also interested in, but I haven't really touched upon today, is we're actively looking at how these tumors actually evolve on treatment, because that's an additional aspect that we also need to consider. So finally, I would just like to thank, this is a big team, obviously. So I would like to thank all the members of my team, all the collaborators on this project, the funding bodies. But I would also like to give a special shout out to Mrs. Jane Hemstrich, who really provided long-term support through a dedicated fellowship to make sure that we can do what we do. We can develop our ideas into real projects and now clinical trials as well. And finally, I would also like to mention the Gergensen Foundation as well, Thomas and Ingeborg Gergensen. Again, we're using some really amazing tech thanks to their support that I didn't talk about today because I promised you imaging. But hopefully I'll talk to you about that next time. So
thank you so much, Marina. Brilliant presentation. And there's so much in there. I think we probably needed at least 2 bite-sized virtual seminars, but you did a really great job in getting through some incredible and very exciting developments that your team and your collaborators are working on. So congratulations for that. We're going to bring up some poll questions now for the audience to vote, and apologies, you will need to get your votes in quickly. You've got 2 separate polls, so you've got the opportunity to vote for one or the other of your favourite questions, and we'll throw those to Marina in just a moment. While we do that, Marina, what's it been like for you with COVID-19? How much of an impact has that had on the acceleration of your work?
Yeah, I mean, certainly.
And Marina, do you mind stopping sharing on your slides?
Yeah. Yeah, it certainly, look, Margine, it certainly affected all of us. But I think, you know, Garvan really had a really good strategy going from the beginning to maximize, you know, our capabilities, to make sure that we can do what we do. So as much as we, particularly, I guess, in the first couple of months, we were slowed down a bit. You know, we also had to go home. We had to tell our people to go home and, you know, slow down the work. Very quickly, though, we were able to amp up and, you know, just continue working. So I wouldn't say I'm more worried about the funding bodies, to be honest, what that landscape is going to look like in the future.
Yeah, absolutely. Good point. I think we've got a lot, and indeed, particularly in pancreatic cancer, because as you've identified, you know, it is such a lethal disease. There are a number of families out in the community doing incredible work with their fundraising, and certainly that's been impacted by not being able to host events or to do walks and so on. So our thoughts are really with them, and I guess medical research, you know, needs needs funding more than ever, I think it's fair to say. So I think we've got the results coming through on the poll. First question for you, please, Marina. What are the current treatment options for pancreatic cancer? And do you see your work having an impact on changing the treatments? I think the answer to that might be yes, based on the clinical trial.
I am so excited. You have no idea how excited I am about this is just one clinical trial, but I think we really have a unique opportunity with, and this is really thanks to David Thomas, but also the whole MOST national program. So all the people that are involved for setting up such a nice infrastructure, nationwide type platform, which is what we need for pancreatic cancer, because to really recruit sufficient numbers of patients for these studies, there are numerous obstacles, in the way to run efficient trials for pancreatic cancer. So I am really enthusiastic. I think that this is just the beginning. We have loads of trials. We have loads of preclinical data in the pipeline. It's almost ready. It's almost at the next stage. So we're almost planning what are the next trials in this area.
That's really exciting. Thank you. Final question, and then we'll have to wrap up. Why is pancreatic cancer typically found when it's so late stage?
Yeah, unfortunately, not many people know what pancreatic cancer, what pancreas does. It's almost like that, actually. Nobody knows what pancreas does until pancreatic cancer is diagnosed. And I guess the problem is that because it is so deep within the body, symptoms often come late. And then when they do come, they're very vague. So, you get things like back pain, you get things like abdominal pain sometimes, and those things could be anything. But I think what's also been great in this space is that there is a lot more awareness now of pancreatic cancer with the public, but also the GPs. So, which is, I think, the important component actually of getting the diagnosis much earlier. So if the GPs are educated and can really start the testing earlier, there's a much better chance of getting this diagnosed earlier.
Fantastic. Thank you so much, Marina. So look, Marina, thank you very much for sharing with our audience today, and everybody in the Zoom room, thank you for your time and for your support and for coming to hear about this really important work. There will be a short survey popping up soon, and we would love your full and frank feedback. There are always opportunities for us to improve, so we want to hear from you on how we can do that and make the bite-sized science seminars even better than they already are. As you've heard from Marina, philanthropic support and community support is absolutely critical, not just to retaining the incredible medical research that we have in this country, including here at the Garvan Institute, but to really accelerating the pace of research findings, but most importantly in supporting our bright scientists such as Marina. And you've heard a little bit today about philanthropic fellowships, which can really make the difference between somebody being able to have a long and successful career in science or really, struggling just with getting the funding together to underpin one's salary. So we cannot thank you enough for supporting Garvan. Please go tell your friends about us. Please encourage them to come along to a seminar, sign up to the Garvan e-news and so on, and do stay tuned for details on the next Bite Size seminar. Thanks, everyone. Have a great day.